Hello students, today we will do the revision work and discuss about all the topics that we have studied in the last classes. First topic of this is what are chromosomes. So what are chromosomes? Chromosomes are the thread-like structures which get condensed during the process of cell division or before the process of cell division. They get condensed, they coiled and form chromatids. Two chromatids, they join together. Chromatids join together at the point of contact, which is known as centromere. So this is about chromosomes. Chromosome is and it contains the genetic material. It is present inside the nucleus. It contains the genetic material or the DNA, which is responsible for the transmission of characters from one generation to another. Which is the discovery of chromosome. Discovery of chromosome, it was discovered by Sir Walter Fleming in 1882. In the larval cells of salamander or dividing cells of larva of the salamander, here he found that the thin thread-like structures are present and he named it chromatin or chromatin, which literally means thread. So the name was given by Walter Fleming, German scientist. This was about the discovery. Then the chromatin. Chromatin material. Chromatin is the genetic material or the material which carries the genetic genes or the DNA segments on it. And chromatin material, it basically contains 40% of DNA and 60% of histone proteins. 60% of histone proteins. 40% of DNA. If we are discussing about the DNA structure, DNA is a double-stranded helix or the coiled helix, which is double-stranded with a sugar phosphate backbone supported by the ladders, which is made up of nitrogenous bases. Nitrogenous bases they are basically adenine, guanine, cytosine and thymine. These two, the pairing of these nitrogenous bases is also A always pairs with T by double hydrogen bond, or adenine always pairs with thymine with a double hydrogen bond. Guanine always pairs with C by a triple hydrogen bond. So this is how the structure of DNA is. This structure of DNA was proposed by Watson and Crick. So double helical structure or the helical structure, double-stranded helical structure supported by nitrogenous bases as ladders. Then the next topic is histone proteins. Histone proteins are the special proteins around which the chromatin fibers are winded or coiled or wrapped. So the histone proteins, we can say, are the special proteins around which the chromatin fibers are wrapped, and they make a group of 8, which is octamer. Group of 8 forms nucleosome. After that, they make a structure or the coiled structure like this. First they form a divalent, which is known as a chromatid. Single one, it is known as chromatid. If two chromatids join together, the point of contact is known as centromere. Next point that we have discussed is genes. Genes are a special type of coding or a sequence of nucleotides. Sequence of nucleotide which is responsible for the transmission of character. So sequence of nucleotides, sugar, phosphate and nitrogenous base. This sequence, the sequence of nucleotide form is known as genes, which is responsible for the transmission of characters. The two strands of DNA strands are complementary to each other. Complementary means if we know the sequence of one of the strands complementary to each other. If we know the sequence of one of the strands, we can easily identify the sequence which is the next. If the sequence is like this, then we know the second one, the complementary strand, it must be this. So this is how we can identify, or we can find out the complementary strand DNA or DNA sequence or gene sequence. Then next is need for new cells. Need for new cells. New cells are required for the growth, repair and replacement. Growth of an organism, repair of the dead and torn out tissues, replacement of cells. New cells are formed because of to remove the dead cells. Continuous wear and tear of cells takes place as in case of skin. For the replacement of dead cells, new cells are all formed regularly. So growth, repair and replacement, all these processes takes place by the mitotic division or mitosis. Then the next is formation of reproductive cells, sperm or ova, or in case of flowering plants, ovule and pollen grains. So this reproductive cells, they are formed by the meiosis division. Next topic for this is cell cycle. Cell cycle is a sequential arrangement of the steps that takes place during the process of cell division or the cell before all the steps, whether it is of the cell division process or the resting stage, altogether the sequential arrangement is known as cell cycle. It is interphase, and then the four stages, which is prophase, metaphase, anaphase and telophase. All these phases are the phases of cell division, while interphase is the resting phase, where the cell prepares itself for the division process. Then next topic is formation of new DNA by the semi-conservative theory. The first step is uncoiling of the DNA strand. After uncoiling, it forms a replication fork and it acts as a DNA template for the formation of new one. So then two new strands are formed, one with old and one with new strand. So this first step was uncoiling. Second is formation of replication fork. Third, formation of new DNA. new DNA, which is semi-conservative, one old and one new strand. This is based on semi-conservative theory. Conservative theory. Next topic is cell cycle go on endlessly. Can the cell cycle go on endlessly? The answer is like brain cells. Once they are formed, they only grow in size. Liver cells they divide once or twice in a year. Then some cells like cells of the skin, it divides continuously to remove the dead cells which is formed during the wear and tear process, or for the repairing of cells. The division process in skin goes on continuously or endlessly. So every cell is not dividing continuously. Some cells they divide once in a year, some cells they divide continuously. So this one. Then mitosis and meiosis. Mitosis is the process which takes place in the somatic cells. Somatic cells which is responsible for the growth, repair and replacement process. While meiotic division it takes place in the reproductive cells. Reproductive cells. To form the sex cells which when fused together during the process of fertilization, they form the zygote, and to retain, then the last topic is the significance of mitosis and meiosis. Last topic of this chapter is significance. Significance of mitosis and meiosis. So first we will discuss about mitosis. Mitosis, thus three only: growth, repair and replacement. Growth of an individual, since the one cell divided into 2, 4, 6, 8 and this which results in the increase in length or the volume of the cell of the body or the organism. Then repairing the torn out or dead tissues, replacement of the tissues which are formed during the wear and tear process. Then meiosis. Significance of meiosis: formation of reproductive cells. Mitotic division is the equational division. Equational division, as the number of cells remains the same as the parental cells, or the diploid number is retained in this step. Diploid number of the parental cell is retained in the mitotic division. So it is also known as equational division. Then in this process it is the reductional division. Reductional division, and the chromosome number becomes N, which is haploid. Half of the parental chromosome number. Half of the parental chromosome number is diploid set, a paired set, but the chromosome number of the egg and ova is haploid, since when they fuse together during the process of fertilization, the diploid number is retained. Diploid number retained in the zygote stage. This is all about this chapter. Thank you.